Question 3. The student was disappointed with the slide that had been prepared because the nuclei were poorly stained and no stages of mitosis could be seen. Explain why the student had to make sure that an appropriate stain was used. So for 1 mark, they're looking for the idea that the stains needed to see the chromosomes that would not be clearly visible if we did not use the stain. They will accept so that you can see the chromatids, also you can see the nucleus. Or you could have, because the stain must attach to the chromosomes or the DNA or the histone, or the idea that the stain must be absorbed or be taken up by the chromosomes or the DNA. Part 2. Describe the changes that need to be made to this method to allow the stages of mitosis to be seen. So when we did our root tip squash in class, we used the very end of the root tip because this is where mitosis is going to be happening the most. So in this experiment they used 2 centimeters from the plant, but we need to make sure that we're taking the very end of the root tip, so we should be using a shorter piece of the root tip, preferably about 5 millimeters long. We added the acid and then the stain separately. In this experiment they would add them together. The acid is to kill the cells so that mitosis will stop, and the stain is then added to see the nuclei and the chromosomes. They will also accept the idea of use a more concentrated acid. If you heat the root tip when it's in the acid and the stain, could also increase those results. When we did our experiment, we used a needle and we macerated the root tip cells before we placed it on the slide. So the idea that if we tease the cells apart before the staining, or we macerate those particular cells, they're more likely to take up the stain. Or we could intensify the stain by warming up the stain after squashing.